and Jai Hind everyone. So I know it has been a while since I have uploaded anything for biology, but now as I am back, I didn't came empty-handed. I came with a little gift for everyone, and that gift would be PYQs or previous year question series for biology, in which I have individually picked out the questions from all the years dating back to last 5 years. All the questions that has came in biology and have compiled it together in form of small PYQ year-wise videos that I'll be uploading one by one. In this series, we will be solving all the previous year questions from the last 5 years, either it is of NDA or of CDS. And I'll be telling you some tips, tricks, how to do questions, how to perform elimination method when something out of the world comes in the examination. I'll also clear some concepts which are required for understanding those questions, and that will definitely boost your preparation for the examination. So that is it. This will be our first lecture of this PYQ series, in which I have included all the questions that have came in CDS 2024 Part 1, CDS 2024 Part 2, CDS 2025 Part 1. All the 30 questions are compiled here. So what are you waiting for now? Let's get started with the series and hope it helps you a lot in your examination. So the first question of our lecture is here present on your screen. That is an antibacterial enzyme that is present in saliva and in tears is called. Now see, this question may be a very abrupt question for everyone, right? In the examination. So what we will do is we won't panic and we will see the options. We will try to eliminate as much options as possible as per our previous knowledge or as per our prior knowledge, whatever we have about biology. Okay, so let's see the enzymes which are given in the options. Our first enzyme, which I see is common to you all, will be lipase. Why? Because lipase is an enzyme which is used to convert fat into fatty acid and glycerol. Okay, and we all know that the primary function of saliva is the digestion of carbs, digestion of carbs or carbohydrates. Okay, so definitely fat enzyme will not be beneficial for this question. Now, one more enzyme that I'm seeing here is isomerase. Now, this enzyme is an enzyme which converts isomers from one form to other. One perfect example that I can think of is during glycolysis, glucose compounds are converted into fructose compounds and vice versa. Now, this change of both are six carbon molecules, but glucose has aldehyde as the functional group and fructose has ketone as the functional group. These both are isomers of carbon 16. Because of that, because of the change of group, these are two different isomers, and the enzyme which is beneficial for that is isomerase. So this again has no relation with carbohydrates. So we will just leave these two. Then understand, we want to find an enzyme which is antibacterial. That means, what do you mean by antibacterial? Which enzyme is anti or against bacteria? That means an enzyme which can kill bacteria, right? So you must have heard about lysosome, if not lysozyme. So lysosome was a cell organelle. Everybody knows about it. It was known as the suicide bags of the cell. And what was the function of lysosome? The function of lysosome was to digest any foreign material which may have come with the within the cell. Okay, so कुछ भी foreign material हो, चाहे वो बैक्टीरिया हो, चाहे कोई और infectious agent हो, ये उसे digest कर देता था. ठीक है, that's why it is anti-bacteria. It may be antibacteria because of the hydrolytic enzymes that are present in lysosome, right? So we can think that Among these two options, lysozyme can be our correct option as per the previous knowledge that we have. And let me tell you, if you have guessed lysozyme, that is the correct answer for this question. So lysozyme is the enzyme which is an antibacterial enzyme present in saliva and in tears. Now let's move on to second question. Second question states that which set of vegetable is an underground stem or modified stem? Now, nobody is expecting you, neither me nor the examiner, कि आपको हर एग्जाम्पल याद होगा, ठीक है? लेकिन कुछ एग्जाम्पल्स जो एनसीईआरटी में है वो आपको पता होने चाहिए, याद होने चाहिए, ओके? One such example in NCERT is turnip and carrot. Both of them are modifications of root for the storage of food. Okay, so these two are root modification. It has nothing to do with the stem. So automatically हमारे दो options that is B and D cancel हो जाते हैं. अब हमारे पास जो दो और options बचे हैं that is A and C, उनमें अगर आप ध्यान से देखोगे तो it has been written in the first option round gourd. Round gourd in Hindi is gourd gobi, and everyone who knows about anything about round gourd or gourd gobi is that it is a fruit, not a stem, not leaf, not root. Okay, it is a fruit. That's why this option, because one option is fruit, this entire option gets eliminated, and the only option that we are left with is potato, onion, and garlic. अब इस क्वेश्चन से आपको क्या पता चला? अब कोई आपसे पूछेगा व्हाट इज द एग्जांपल ऑफ मॉडिफाइड स्टेम, यू विल से पोटैटो, अनियन एंड गार्लिक. दीज आर दी थ्री एग्जांपल्स. ये डायरेक्टली भी पूछ सकता है आपसे. क्वेश्चन ऐसे भी आ सकता है विच अमंग द फॉलोइंग इज नॉट ए मॉडिफिकेशन ऑफ स्टेम, एंड इट विल गिव यू दीज थ्री ऑप्शंस, एंड वन ऑप्शन कैन बी सपोज कैरेट. तो नाउ यू रिमेंबर दैट दीज थ्री तो आर मॉडिफिकेशंस ऑफ स्टेम. जो एक नहीं होगा वही ऑप्शन आपका करेक्ट हो जाएगा. दैट इज हाउ क्वेश्चंस आर फॉर्म्ड. नाउ थर्ड क्वेश्चन इज विच सेल इन ह्यूमन हैज नो न्यूक्लियस. अब हमारे टाइम पे मैं तो इसको फन फैक्ट की तरह याद करती थी दैट द सेल विच डू नॉट हैव न्यूक्लियस इज नोन एज आरबीसी. क्यों नहीं होता? आई वॉज वेरी क्यूरियस व्हाई RBC has a biconcave structure. Biconcave is a structure which looks like donut or balushahi in which there is a dip in the middle. Okay, that dip in the middle is because there is no nucleus present there. Why that happens? When the RBCs mature, their main function is the transport of oxygen. Okay, so transport of oxygen becomes its main function, and to increase the surface area for binding of oxygen, the nucleus get vanished from the RBC. That means the young RBCs have nucleus, and as soon as they mature, because of their primary function of transportation of O2, they have to increase the surface area for binding. That's why nucleus is uh, vanished from the mature RBCs. The answer to this question is that's why RBCs. Okay. Now the fifth question or the fourth question of the slide is the use of plastic has led to many environment related problems. Okay. Now which one of the following statement is correct regarding that? ये तो basic common sense का question है that plastic, because of the plastic consumption, it is leading to environmental problems. अब related to plastic, which statement is correct? किसी को नहीं भी पता है तो ये तो पता होगा plastics are non biodegradable. वो environment में degrade नहीं होते हैं by the decomposers. They do not decompose. They just get accumulated for millions and millions of years. So automatically you must think that option number A is correct, and if you think that, it is very true. Option number A is correct. That is plastics are non biodegradable. That's why they are causing environment related problem, because if there will be a pile of plastic, it will go into water bodies causing water pollution. If you will burn the plastic, it will release poisonous gases. Because of that, it is an hazard to air, causing air pollution. Then it is choking hazard to cattle, and so many more problems are there. If they get accumulated, they also um, lead to land related pollution, soil pollution, and land pollution. So, so many environment problems are related to plastics. Why? Because they are non biodegradable and they cannot be decomposed by the decomposers. So, the answer to this is option number A. Then the last question on the slide is microbodies found to be present in plants and animal cells contain. Up this is fact-based question. If you have heard about cell organelles, microbody is a type of cell organelle only. It is a membrane-bounded cell organelle present in eukaryotes or eukaryotic cells, and it is composed of, its composition is of enzymes. So the answer to this particular question is enzymes. Okay. Now let's move on to the second slide. First question of this slide is pulse is felt due to rhythmic contraction and relaxation of. Now, let me tell you, our heart has four chambers. There are upper two chambers and lower two chambers. Upper two chambers are known as atria or atrium, whereas the lower chambers are called ventricles. And the pulse is generated due to the contraction and relaxation of ventricles. Okay. And these ventricles, because ventricle is not in the option, these ventricles are directly connected to aorta and major arteries. Okay. All the arteries and aortas are directly connected to it. So whenever we have to sense the pulse, we check from the either from the aorta or the major arteries, and there we can feel the rhythmic contraction and relaxation of pulse. That's why the correct option to this question is C, that is aorta and main arteries, where we can feel the sensation of pulse. Now the next question is when eggs are heated, the transparent liquid portion around yolk turns solid and turbid white. Now this happens due to the thermal denaturation of, well, the answer to this is protein. Let me tell you why. This liquid portion, white colored portion around the yolk is a protein known as albumin. Now this albumin protein has a 3D shape, or it is a 3D protein. Now proteins can be primary or linear. They can be secondary, tertiary and quaternary as well. And as they increase from primary to tertiary, what happens is that in the linear uh, protein molecule, coiling is happens, and a lot of folding happen. Because of that, ultimately the protein becomes a 3D structure, three-dimensional structure, and that is what we call 3D proteins or tertiary proteins. Now, albumin is a type of tertiary protein, and whenever these proteins are given heat, excess heat, then they denature. What do you mean by denature? They lose their 3D shape and again will form linear chains of protein. They will turn back to linear chains from 3D structure when heat is given to them. When excess heat is given to them, this denaturation happens from 3D to linear chains, and that is why albumin gets turbid white and solid when we provide heat to it or when we boil the eggs. So the correct option is denaturation of proteins. Now question number 62. The causal organism of dengue fever is mosquito-borne virus which belongs to which one of the following categories? Now, this तो question में ही दे रखा है that dengue fever is caused by mosquito-borne virus. That means the causal agent of dengue fever is a virus, whereas the vector by which dengue fever is caused is a mosquito. Now I'll tell both the specific names of the virus as well as the mosquito. Mosquito called Aedes mosquito is the responsible vector for dengue, whereas the causal virus is a virus of class flaviribo. That means the virus is a flavivirus or flaviribovirus. Okay, so the correct option is A. Now let me tell you about the other viruses as well. It may happen that the options can be repeated. Okay, so adenovirus is usually responsible for all the let's say lung related or pulmonary related issues, as well as gastrointestinal problems as well, diseases as well, uh, s- diseases. Okay. Apart from that, I'll give you the symptoms of adenovirus can be cold, can be pneumonia, can be cough. Okay. So these are the diseases that can happen with the help of or due to adenovirus. Then the next virus given is vaccinia virus. As the name suggests, this virus is used in the vaccine of smallpox. So please remember that. And there is Nipah virus. This is a virus that is related to bats. Now, COVID was very famous in 2019 and 2020, right? Why? Because somebody in Wuhan ate the bat. Because of that, COVID was spread as a pandemic. So that's why this virus was even in the option because it is related to bats. So if you eat the fruit contaminated by the bats or the water inhabited by the bats, this virus is more likely to occur. So that's why all the viruses are given, and I have given you like why this virus is used or due to which this virus can occur. So please remember that. Now the last question of the slide is, from which of the following are blood clotting factors released? Okay. First of all, what you should know about blood is that blood is made up of two components. One is plasma, other is blood corpuscle. Okay. Now blood corpuscle has three parts, that is RBCs, WBCs, and platelets. I'll give you main functions of all the three. RBC is used in the transport of oxygen. WBC is the immunity of our body. That means it fights with the foreign infectious agents. Platelets are the ones which are involved in forming clot. Whenever we get an injury, you will see a thread-like, net-like structure, which is brown in color, appears over the injured wounds. Okay, that is the function of platelet, so that the excess blood loss can be prevented. So, for this question only, the correct answer of the blood clotting factor would be platelet. But one more thing I'll tell you here only is that questions on WBCs are given a lot. For example, these two options, if you see eosinophils and monocytes, these are the types of WBCs. I have seen some question in India also, which has come from which of the following is a type of WBC. So we will just learn about types of WBCs in 1 minute. Okay. So WBCs are divided into two parts, granulocytes and agranulocytes. Okay, granulocytes have a grainy appearance, whereas agranulocytes do not have that appearance. Granulocytes are further divided into three. I call it Ben. सबने Ben 10 देखा है ना? That cartoon. So I call it Ben. Why? Because we have three granulocytes, and these are basophils, eosinophils, and neutrophils. Okay. All these come under WBC. And in agranulocytes, we have two. They are monocytes and lymphocytes. Okay. Then again, the lymphocyte is further divided into two. That is B lymphocyte and T lymphocyte. That is all that you should know about types of WBCs. Okay. So please remember this because this is the new favorite question of the examiner, WBCs. Okay. So that is it for this slide. Let's move on to the next slide. Now, in the next slide, our first question is photo 51 refers to an image of. Now, it may happen that you don't know about photo 51. To be very honest, I also didn't know about photo 51. But if I have to solve this question, I will go through elimination even if I don't know the answer. Why? Because I'll tell you in a short while. First of all, I know that the virus responsible for common cold is definitely not related to photo 51 because the virus is called rhinovirus. Okay. So I haven't heard anything related to photo 51 with rhinovirus. Okay. Then the virus responsible for COVID is SARS, I think, SARS-CoV-2. That virus is also not related to photo 51. And this much current affair I know that photo 51 is not related to any crater on the moon. So only option I am left with is DNA molecule. So my best bet would be it is something related to the image of DNA molecules. Okay. And the correct option would also be the DNA molecules. Why? Because photo 51 is the X-ray diffraction image of DNA molecule. Image of DNA molecule only. Okay. So this is the correct option. Again, I'm telling you, if you think that question is very new, then please understand अगर आपको नहीं आ रहा तो बहुत लोगों को नहीं आ रहा होगा. The best possible bet that you can take if you want to take it is that आप options देखो, जितने ज्यादा eliminate कर सको उतने eliminate करो. If you reach to the perfect conclusion, then go ahead with it. If you don't feel confident, leave it. If you don't know, people like 80-90% people would not be knowing this. Okay. No worries during the examination. Be stress free and exam will go absolutely fine. Now, the next question, the instrument which is used to measure the blood pressure manually is called. Well, the answer to it is sphygmomanometer. Now, let me tell you from the blood pressure, 3-4 questions, 2-3 questions are asked. First, I have seen the question on what is the normal blood pressure, okay, of a human. Now, if you know the answer to it, please write in the chat box, okay? And I'll like the comment which is correct, okay? Then the second question could be about high blood pressure. How much blood pressure is considered to be a high blood pressure, okay? And the third question is about the instrument that is used to measure the blood pressure manually, and the answer to it is the device which has particularly long name, that is sphygmomanometer. So that is the correct option to this question. Now, the next question states that why does rough endoplasmic reticulum looks rough under a microscope? Now, if you know anything about ER, that is endoplasmic reticulum, is that ER is divided into two types. One is RER, that is rough endoplasmic reticulum, and other is SER, that is smooth endoplasmic reticulum. Now, the only distinction between SER and RER on the basis of physical appearance is that RER has a rough appearance because of the presence of ribosomes that are formed in the nucleolus and then get transferred to the surface of ER, forming RER or rough endoplasmic reticulum. So R for rough and R for ribosome. So if the ribosome get attached to the surface of endoplasmic reticulum, it becomes RER, And because of this ribosome, it will synthesize protein. And the SCR, because the ribosome is not present, it will not synthesize protein. Rather than the synthesis of protein, it will be involved in lipid synthesis. So that is all about ER. And the answer to this, it has a rough appearance because of the presence of ribosomes that are attached to its surface. So first option is correct. Now the last option of the last question of this slide is, how many carbon atoms are present in pyruvate molecule? Now nobody will say कि ये question बाहर से आया है, because if you have heard anything about glycolysis, this is a process which you must have heard in respiration. Okay, so glycolysis is the first step towards aerobic as well as anaerobic respiration. Now what happens in glycolysis? We have glucose molecule in glycolysis which gets converted into pyruvate or pyruvic acid. Okay, they both are same thing. So 6 carbon glucose gets converted into 2 molecules of 3 carbon pyruvate during glycolysis. This is the process, this is the first step towards anaerobic or aerobic respiration, and this is important. That's why you need to learn. In glycolysis, glucose is converted into pyruvate. Since glucose is a 6 carbon molecule, it breaks down into 2 molecules of pyruvate, each of 3 carbon atoms. So the correct answer to this is 3. 80 mmHg in a normal blood pressure. Okay, please understand this. In normal blood pressure, 120 is systolic pressure, 80 is diastolic pressure. Now you must be wondering ये question कहाँ से आ गया? So if you will see the circulation in human beings, there is a do you know chart, yellow colored box on the uh, top of the page, where it has been written about systolic and diastolic pressure, where it has been written about normal blood pressure of human and high blood pressure of human beings. Okay, so go through it. Everything in NCRT is important, especially the boxes which indicates did you know, fun fact, and things like that. Okay, chalo, let's move on to the next slide. So now let's come to this slide. अभी जोश डाउन नहीं करना है. Only 2 slides or 3 slides are left. Keep your जोश up. हो जाएगा. So let's move on to the first question of this slide. Which one among the following is not a micronutrient for the plant? Now, plant for their growth and development require nutrients, and these nutrients cannot can be classified into macronutrients and micronutrients. Now, what are macro and micronutrients? Macronutrients are the nutrients which are given in large quantities or which are required in large quantities by the plants, whereas micronutrients are required by the plants in trace amounts, that is very small amounts are required by the plants. Now if you want to do this question right, all you have to do is either learn all the macronutrients or learn all the micronutrients. So I'll suggest just learn the macronutrients. Why? Because NPK, जिसका भी गांव में घर है उसे पता होगा, जिसने भी फार्मिंग वगैरह देखी हुई है, कि एनपीके इज अ फर्टिलाइजर विच इज वेरी नेसेसरी फॉर प्लांट्स. और जब भी हम लोग फसल वगैरह उगाते हैं तो हम लोग एनपीके छिड़कते हैं. दैट फर्टिलाइजर ऑलवेज कम टू आवर होम्स. सो एनपीके are the 3 macronutrients which are important for the plants, that are nitrogen, potassium, and phosphorus. Okay, apart from that, Ca, Mg is also important. Then sulfur, carbon, and hydrogen is also important. So these are the macronutrients. अब मुझे याद कैसे हुआ था वो मैं बता देती हूँ. NPK कार मांगे सेठ चोर है. That is how I learned. This is called mnemonics, and you can make a mnemonics of your own, but that's what worked for me. because that's what my teacher made me learn. So this is how you learn all the macronutrients which are necessary for plants, that is nitrogen, potassium, phosphorus, calcium, magnesium, sulfur, carbon and hydrogen. Okay. Abhi, if you will see here, magnesium is a macronutrient and all the three are micronutrients. So if we have to choose which is not a micronutrient, then it will be magnesium because it is a macronutrient. Fine. Now let's come to the second question. The xylem in plants are responsible for the transport of. ये बहुत simple question है. If you have learned anything about complex permanent tissues, then you must have learnt about xylem and phloem. There are only two complex permanent tissues, that is xylem and phloem. Now, in phloem, the sound of fur happens, and in food also the sound of fur comes when you start the word. So, phloem is connected with food conduction, or we call it translocation of food, and xylem is involved with the conduction of water and minerals. So that is very basic question. Xylem it helps in the transportation of water and dissolved minerals, whereas phloem is there for translocation of food. That means transport of food from source to sink. Okay. Now the third question is, which one among the following hormones inhibits the growth activity? लो ये तो वो वाली बात हो गई मैंने अभी बताया था वाली. यहाँ पे मैंने प्लांट हार्मोन सारे लिखे हैं ग्रोथ इनहिबिटिंग हार्मोन्स में जो प्लांट की ग्रोथ को रोकते हैं दैट इनहिबिट्स द ग्रोथ ऑफ प्लांट आर ओनली टू. दीज आर एब्सिसिक एसिड एंड इथिलीन. तो हम लोग चेक कर लेते हैं ऑप्शन में ये दोनों है या नहीं है. एक है दैट इज एब्सिसिक एसिड, वही आपका करेक्ट ऑप्शन हो जाएगा. इसका एब्रीविएशन इज ABA विच आई हैव रिटन. And rest auxin, cytokine and gibberellin. These are also plant hormones, but they are not inhibiting. They are growth promoting. That means they are growth promoting plant hormones. Okay, all the three. Now the next question. Halophytes are the plants that grow on which one of among the following categories of soil? So halo is a word that comes from saline. That's why the plants which are halophytes are actually grown in saline soil. Okay. So again, that is a fact-based question. This is a question which you can also relate somewhere with geography. If you have read a little bit of geography, you should know that halo is related to saline. It will give you an idea about what to mark in the question paper if you want to have a bet. Now the last question on the slide is, which one among the following is not a part of vascular bundle? चलो अब vascular bundle आपको बता ही देते हैं. If you know, if you don't know, still. So vascular bundle has three parts. Okay. The center or the middle part is known as cambium. Outer part is phloem and the inner part is xylem. These are the three parts of the vascular bundle. Anything except these three parts is not a part of vascular bundle, and here in the option it is endodermis. दिख रहा है या नहीं दिख रहा है? अगर नहीं दिख रहा है, let me erase this. Okay. So the answer to the last question is endodermis. अगर आपको कैम्बियम नहीं वैस्कुलर बंडल नहीं भी पता है, then also you know that dermis means the outer layer of any organism. In animals it is skin, in in humans it is skin, in plants it is the outer layer. Okay, so derma is related to skin basically. So this cannot be a part of vascular bundle. That's why this option is again correct. Chalo, let's move on to the next slide now. Okay, so the first question on this slide is, which one of the following category of biomolecules does starch belong to? नाउ अगर आप पीछे जाओगे तो मैंने आपको कार्बोहाइड्रेट्स बहुत अच्छे से एक्सप्लेन किया, उसमें मैंने आपको बताया था स्टार्च इज अ टाइप ऑफ पॉलीसेक्राइड, ओके। सिंस इट इज अ टाइप ऑफ पॉलीसेक्राइड, इट इज अ टाइप ऑफ carbohydrate. So starch is a polysaccharide which belongs to the biomolecules called carbohydrates. So the answer to this is carbohydrate. Then which one among the following is not a feature of xerophytes? Now it is, first of all, please, it has been bolded that you have to tell me which is not a feature of xerophyte. पहले इसके लिए तो जानना पड़ेगा xerophyte है क्या. So xerophytes are the plants which live in arid conditions. Now what is arid conditions? These are the conditions where there is no to um, trace amounts of water. जहाँ वाटर बहुत कम अमाउंट्स में होता है, टेम्परेचर बहुत ज्यादा होता है. फॉर एग्जाम्पल जितने डेजर्ट वाले इलाके हैं, दे इन दीज कंडीशंस इफ अ प्लांट इज ग्रोइंग दे आर कॉल्ड जेरोफाइट्स. अब जेरोफाइट्स क्योंकि वहां पे पानी कम है, डेजर्ट में पानी है ही नहीं और बहुत कम है, इसकी वजह से जो प्लांट्स होते हैं उनमें एडेप्टेशंस होती हैं, और ये एडेप्टेशंस इस तरीके से होंगी कि पानी बचाया जाए. ठीक है. अब पानी बचाने के लिए क्या चाहिए होगा? लीव्स पे है स्टोमेटा, स्टोमेटा के थ्रू होता है ट्रांसपिरेशन, दैट मीन्स वाटर लॉस होता है थ्रू स्टोमेटा. अब पानी बचाना है तो लीव्स को पहले तो छोटा करना पड़ेगा ताकि स्टोमेटा कम हो जाए. इसके अलावा ये भी कर सकते हो, लीव्स के ऊपर एक वैक्सी कोटिंग आ जाए, क्यूटिकल की कोटिंग आ जाए, जिसकी वजह से बिकॉज ऑफ द वैक्स डिपोजिशन वाटर लॉस कम हो. और क्या हो सकता है? स्टोमेटा प्रॉपर एक्सपोज्ड होने की जगह पे डीप डाउन संकन हो इन दी लीव्स, तो भी हमारा वाटर लॉस कम होगा. सो ये तो तीन फीचर्स हैं जो जेरोफाइट्स के होने चाहिए. सिर्फ एक फीचर विच इज प्रेजेंस ऑफ लार्ज नंबर ऑफ स्टोमेटा इज नॉट ट्रू. अब लार्ज नंबर ऑफ स्टोमेटा होगा तो पानी लॉस दिन भर होता रहेगा. That is not beneficial for xerophytes. That's why this option is the correct option, and this is not a feature of xerophytes. So our correct option would be B. Now, question number 3 on this slide is which organelle in plant cell can digest foreign material? अब तो जिसने स्टार्टिंग से ये लेक्चर देखा है उसे पता है कि the cell organelle which can digest any foreign material is the one which we call suicide bags. तो अभी के लिए मैं इसका आंसर नहीं दूंगी. Everyone who know the answer, please write it in the chat box. चलो, let's move on to the next question. Photosynthesis that converts light energy into chemical energy by plants involves the use of green colored pigment of leaves called chlorophyll. This is the statement of the question. Now the question starts from here. The only structure in leaves that host chlorophyll is. Host chlorophyll means that contains chlorophyll. Now there is a cell organelle which is known as plastid. Okay. And this plastid is further divided into three components. One is chloroplast, जिसमें क्लोरोफिल होता है, और ये हमारे क्वेश्चन का करेक्ट आंसर भी है. अदर इज क्रोमोप्लास्ट. It is responsible for all the colors, all the bright colors of petals and fruits, like tomato red color, brinjal purple color, and other colors also. This chromoplast is responsible for those colors except green color, which is the property of chloroplast because of the chlorophyll present in it. And the last one is leucoplast, which is transparent in color. Question of this lecture is question which states that which of the following statement is are correct about vaccination that provides protection against an infectious disease. So last question है, सब ध्यान से सुनो. वैक्सीनेशन, वैक्सीनेशन के लिए इंपॉर्टेंट है वैक्सीन. अब वैक्सीन होती क्या है पहले तो ये बताती हूँ. वैक्सीन इज अ प्रिवेंटिव मेजर अगेंस्ट एनी इन्फेक्शियस डिजीज. अब प्रिवेंटिव मेजर होता क्या है? जिस भी डिजीज के लिए वैक्सीन है वो डिजीज होने से पहले लगाई जाती है। जैसे पोलियो की जो वैक्सीन्स लगती हैं वो पोलियो होने से पहले लगाया जाता है, छोटे बड़ी बच्चों में लगा दिया जाता है। क्यों? क्योंकि जो वैक्सीन्स होती हैं वो एक्चुअल में क्या है, वैक्सीन्स आर नथिंग बट vaccine has dead and weakened, dead or weakened microorganism of the same disease. Okay, microorganism of same disease. For example, पोलियो है तो पोलियो की वैक्सीन में क्या होगा, डेड एंड डिकेड और डेड एंड वीकेंड माइक्रो ऑर्गेनिज्म ऑफ पोलियो डिजीज। ओके, जो माइक्रो ऑर्गेनिज्म पोलियो डिजीज कॉज करते हैं उन्हीं का वीकेंड वर्जन हमारी वैक्सीन में होता है, जिसपे हमारा इम्यून सिस्टम एक्ट करके फाइट करके इजीली जीत सकता है जिससे। ठीक है, सो जो वैक्सीन होती है वो डेड एंड वीकेंड ऑर्गेनिज्म से बनी होती है, सेम डिजीज के। दैट्स व्हाई when the vaccine is given to the animal or human, what happens is that these vaccines with these organisms get inside the body produces antibodies to fight with the diseased organisms, microorganisms. With this fight, the antibodies win over this weakened disease causing organism and develop a memory. अब मेमोरी कैसे डेवलप होती है, आपने एक बार लड़ लिया, सारी एंटीबॉडीज को ये पता चल गया कि हमें अगर ऐसा कोई इन्फेक्शियस एजेंट आया तो उससे लड़ना कैसे है। अब नेक्स्ट टाइम जब सेम इन्फेक्शियस एजेंट बॉडी के अंदर आएगा और बॉडी को कंट्रोल करने की कोशिश करेगा तो एंटीबॉडीज और अच्छे प्रेशर के साथ और अच्छे पावर के साथ उससे लड़ पाएंगी एंड अगेन दे विल विन ओवर द रियल इन्फेक्शियस एजेंट एज वेल। सो वैक्सीन का तो ये काम होता है। अब ऑप्शंस देख लेते हैं जो गिवन है। पहला, वैक्सीन इट ब्लॉक्स द एंट्री ऑफ इन्फेक्शियस एजेंट इनटू द बॉडी। नहीं, वैक्सीन ब्लॉक नहीं कर सकती है, वो एंटीबॉडीज जनरेट करती है उससे फाइट करने के लिए, उस इन्फेक्शियस एजेंट से फाइट करने के लिए, बट वो ब्लॉक नहीं कर सकती एंट्री। ठीक है, सो पहला ऑप्शन इज रॉन्ग। सेकेंड, इट प्रोड्यूसेस एंटीबॉडीज अगेंस्ट द इन्फेक्शन, एब्सोल्युटली राइट, अभी बताया है। Then the third option is it kills the infectious agents when it enters the body. नहीं, वैक्सीन किल नहीं करती है, वैक्सीन एंटीबॉडीज प्रोड्यूस करती है, एंटीबॉडीज किल करती हैं इन्फेक्शियस एजेंट्स को। वैक्सीन का डायरेक्ट काम इन्फेक्शियस एजेंट्स को किल करना नहीं है। सो दिस इज ऑल्सो रॉन्ग एंड द ओनली राइट ऑप्शन इज सेकेंड ओनली। With this, we came to the end of the first lecture of PYQ series. I hope you enjoyed it. I hope these 30 questions are very clear to you. If not, again, see this video again and again for revision purposes so that all the answers are pasted in your memory, जैसे antibodies की memory में paste हो जाता है। ठीक है? With this, I'm ending this lecture and we will see you in the next PYQ series video. Okay, Jai Hind and keep your preparation up. All the best.